Biological membranes. Membranes are designed to be nearly impermeable barriers. We don't want things to get through. That's sort of their function, right? We want to separate things that have different functions. Membranes establish discrete compartments within cells to maintain gradients of protons, gradients of ions, whatever signaling molecules need to be in certain organelles. Okay? So this figure here is kind of showing, just really basically, you have two sides of a cell membrane or a membrane around an organelle. Imagine this continues. It isn't cut off here. There's some continuation to this overall structure. So you might want to be keeping positive charge on one side and negative charge on the other. And so getting molecules across the membrane would incur a huge energetic barrier because we have to push something charged through that hydrophobic region. Okay? So there's an energetic penalty to moving a polar molecule through the hydrophobic layer. You would need to, if you think about it on this end, these groups here that have some sort of polar charge are able to interact with water. So we're going to break those interactions, which takes energy in order to move them through. They won't be able to interact in here. Okay. So exchange of molecules and across the membrane is an essential cellular function. We need this to happen. And so one way that we can facilitate this is some proteins that we embed in our membrane that can facilitate this transfer of these polar molecules for us. Okay. Okay. So membrane proteins will have integral membrane proteins. They're used for both transport and signaling across the membrane. They typically have sort of 2 or 3 key domains. We'll have the segment that actually traverses the membrane, and that part of the protein is going to have a lot of the hydrophobic amino acid residues, okay, because it needs to interact with the tails. So that follows for us. And then we typically are going to have some peripheral domains. And so these are going to be generally populated with a bunch of hydrophilic or polar residues so that they can interact with the aqueous environment either on the outside of the cell or polar molecules on the inside of the cell. And so I just want to kind of point out there's a lot of different orientations for these things. So you can have these barrels here illustrating alpha helices. So the number that you have traversing the membrane can change from these are oversimplified. Generally, when you have bigger proteins, you're going to have way more. Okay, you could have, remember I said arrows are going to represent our beta sheets in our models of the proteins here. So you might have beta sheets are what form the transmembrane portion. And then we have these polar regions, the N-terminal and C-terminal, which side is on which side of the membrane can change. It all just sorts of depends on which protein we're looking at. And there's a lot of possible orientations. Helices are much more common for transmembrane proteins than beta sheets. So if you were asked, what's more likely? Definitely alpha helices. Okay. So again, in order to span the hydrophobic interior of the membrane, the protein segments are going to have to form regular secondary structures to get across, right? Just as they would do in the hydrophobic core. Why? Why do I need an alpha helix or a beta sheet to get across the hydrophobic part of the bilayer? How come I just can't have like just a straight line of amino acids? Yes, great. Perfect, right. So you have these hydrophobic side groups pointing out, and what's on the inside of that alpha helix? If the side groups are what is going to interact with the chains, what makes up the helix? What's in it? It's hydrogen bonds of just the backbone. So the reason we have to have secondary structures is because the backbone needs to make hydrogen bonds. And we said that the backbone is polar, right? Our newly formed peptide bond has a hydrogen bond acceptor and donor. And so we said that alpha helices or beta sheets form to satisfy hydrogen bonding of the backbone. And the alpha helix is going to form hydrogen bonds with itself. Okay? So that's sort of what's shown here. All these yellow dots are our hydrophobic side groups, but the backbone is currently making hydrogen bonds with water. And in order to keep making the hydrogen bonds it needs, in order to get across the membrane, if it's in an alpha helix, the backbone can satisfy H bonds with itself. All the hydrophobic side groups can interact with the tails. Okay. So it's important to remember that the backbone is polar and that both alpha helices and beta sheets both form to satisfy H bonds with the backbone itself. Okay. So this is just another sort of illustration of what that might look like. And then here, because the book codes this, all our polar groups is like red, green, or blue, they're going to be generally on the outside. Okay? So this is key principle for protein biochemistry. Sequence determines what structure it can be in. Structure determines function. Yes? Absolutely. Mm-hmm. No. That's a great question. So he was asking if the dimension of this alpha helix here is big enough to have ions pass through it. And no, the reason this is drawn is sort of like several of these, or this almost looks like a barrel full of beta sheets, is so that you can create a hole large enough for molecules to pass. And so an individual alpha helix traversing the membrane is probably not a transport protein. That's too small. It's not going to transport anything, a single helix. So great.